We have a very special occasion. I'm going to make about 3 quick announcements, and I'm going to introduce our speaker today. But for the people using the department web portal, the members of the department, Sarah mentions that Dr. Mosian's previous lecture was on an overview, an update of epigenetics, which is very much related to the recent work that Dr. Turecki's been doing, and that Dr. Mosian's epigenetics lecture is up on the web portal if you want to review that. That's one thing. Just an update on the residency program. We're going to be submitting our final ranking for the match, and the results of that will be known in March, so that's upcoming. For those of you who missed the February 1st youth conference on resiliency and protective factors against suicide, that DVD will be available soon. If you missed that conference and want to see parts of it or all of it, you can get a copy of that DVD from Emily or Sarah. And then the Grand Rounds today is actually presented in part by donations from the American Foundation for Suicide Prevention, and the Western Mass Chapter, the Department of Public Health Suicide Prevention Program, and Berkshire Medical Center. So what we're going to do today, I'm about to introduce Dr. Turecki, who's going to speak with us. And before I do that, I want to mention that afterwards, Dr. Turecki is one of those rare individuals today who is both a frontline researcher with an extraordinary background in published research, and he is also the director of a mood disorders program, and so he's a clinical leader as well. He's kindly agreed to stay for half an hour after the presentation for those of us who have a little bit more time to talk with him, and I'd really urge you to avail yourself of that opportunity, because it's rare that we find anyone who's able to wear those 2 hats so well, and he's here, and then he's got to get his car to pick him up and get him back up to Montreal. So I'll introduce him now. Dr. Turecki has an MD and a PhD. He's a professor of psychiatry and human genetics at McGill University, where he leads a research and clinical career focusing on depression and suicide. Dr. Turecki obtained his medical degree 1989 and specialty in psychiatry 1994 from UNIFESP São Paulo, Brazil. And I was curious about that because I had lived in Brazil for a couple years when I was younger, and he tells me that his family was to Argentina, and then when he was 10 years old, moved to São Paulo and lived there pretty much until he was 25. And now he's relocated to Canada and McGill. He also got his PhD in neurosciences with a concentration in genetics from McGill University in 1999. He currently holds the William Dawson Chair and is the director of the McGill Group for Suicide Studies and the Quebec Suicide Brain Bank. In addition, he's the Vice Chair of Research and Academic Affairs of the Department of Psychiatry at McGill. Dr. Turecki's research is focused on the study of biomedical risk factors for suicide and depression. He's made important contributions to the understanding of the suicide phenotype, conducting multidisciplinary studies on suicide completers, as well as molecular studies, aiming to understand the suicide brain. More recently, his work is focused on epigenetic mechanisms and has conducted pioneering research leading to our understanding of how early life adversity stably impacts the genome and increases the long-term risk for suicide. Dr. Turecki has authored over 250 publications, including book chapters and research articles in peer-reviewed journals such as Nature, Neuroscience, Molecular Psychiatry, Archives of General Psychiatry, among others. And he is the recipient of several scientific awards, including the NARSAD Michael Kaplan Investigator Award, the American Foundation for Suicide Prevention Distinguished Investigator Award, and the Radio Canada Researcher of the Year 2009. He also serves or has served in the advisory boards of several scientific journals. In addition to this busy scientific career, he is an engaged clinician heading the Depressive Disorders Program at the Douglas Mental Health University Institute, where he treats patients with refractory major depression. So it's really special to have him here. We also want to thank Linda Shippey for all. Why don't you stand up for a minute? Linda brings us a lot of support from AFSP. Thank you, Linda, for helping us get Dr. Trecky going. And so again, for those of you who are coming in the last few minutes, Dr. Trecky will stay after the grand rounds from 1:30 to 2 for a smaller group of us who would like to pick his brains or learn a little more from him before he heads back to McGill. So without further ado, we welcome Dr. Trecky. Thank you.
Thank you. So, hello everyone. It's really a pleasure to be here. I want to thank Linda. It's been fantastic. Thank you very much. How can I turn on the presentation?
Was it up in mid?
Oh, there you go. Okay. Yeah.
Do you want to get in the PowerPoint?
It's down here probably.
You can't see where it is. Yeah.
Oh, okay.
Okay?
Yeah, there you go. Okay, so what I'd like to do is talk a little bit about the work that we do related to suicide. And I'd like to do this informally, so we're not a lot, so please interrupt me if you have questions in the middle about the work that I'm going to discuss with you. So just have nothing to disclose. All right. So suicide is clearly a complex phenomenon, yes, that we believe interact or results from the interaction of a number of different factors. So the way we think about suicide is that people who die by suicide, on one hand, often have factors that increase predisposition and thus that act more distally, increasing predisposition, and then have also factors that precipitate a suicidal crisis, so act more proximally, for instance, like being depressed, feeling hopeless, etc. Although most of the individuals who die by suicide do not have a history of early life adversity, a significant minority does. So about 30% to 40%, depending on the study, have a history of early life adversity, for instance, like childhood maltreatment. What I'd like to do with you today is discuss a little bit of, first, what is the evidence that supports this association between a history of adversity early on in life and increased suicide risk. Then spend most of the time of the talk discussing with you about what are the possible molecular mechanisms that explain this increased risk. And then in the last, for the last few minutes of the talk, if we have time, discuss with you about how is that these molecular changes may translate at the clinical level by increasing suicide risk. All right? So is it okay if I only focus on this one? Yeah? All right. So there are several lines of evidence that suggest an association between having been exposed to adversity early on in life and increasing the risk of suicidal behaviors later on. So this is, for instance, one is from a study in New Zealand. It's from David Ferguson, who's been following a cohort is representative of the general population there, where they see that children that were either sexually abused or physically abused when they turn 16, 18, they are more likely as opposed to those that were not of attempting suicide. This is from our own data from a similar cohort that's representative of the general population from Quebec, and I'm going to discuss with you a little bit more of this work later on. So this a cohort that, again, what we see is that those individuals that were exposed to either physical abuse, sexual abuse, or a combination of both, as compared to those that were not, are more likely, and these at the age of 20, around 20, 21, of manifesting suicidal ideation and suicide attempts. Now, when you go retrospectively, yes, and look at individuals who died by suicide, and this is also from our own data, we see that, again, individuals that die by suicide are more likely to have a history of childhood maltreatment as compared to both psychiatric controls as well as community controls. So clearly, I mean, I could go on and show you data. There's a lot of data that supports this association. I'm sure that, you know, I'm not telling you something you don't know already. Yes? What we know is that it's not the impact of the, or the physical impact of the abuse, but rather the psychological consequence of the abuse that seems to play a role, as suggested by this graph here. This is work in the same cohort from Quebec that we've been studying, where you see that the identity of the abuser moderates the relationship between having been abused early on in life and manifesting suicidal behavior. In other words, that it is important who is the perpetrator of the abuse. So it makes a difference if someone who is close to the family as opposed to someone who is not. Yes, so suggesting that indeed is something to do with the psychological impact, and perhaps, you know, an impact on attachment, an impact of some of the mechanisms that these relationships usually provide. So what I would really like to discuss with you is that how is that this can happen? What happens, yes, that on individuals that were exposed to traumatic experiences early on in life, how is that these traumatic experiences early on in life may increase risk of suicidal behavior many years later? Many years later. So this is the question, you know, that if you, at the end of the talk, this is clear, you know, I'd be happy. So, you know, experience has an important impact in our brain, yes? So, the brain reacts to experience, and we call to this process brain plasticity. Now, the brain is not equally receptive to experience throughout life. There are periods in life in which the brain is more receptive and adapts. There is more brain plasticity according to experience. Yes, and we call to these periods as sensitive periods. So sensitive periods are periods in which our brain is more receptive to adapt and learn with the experience. So this is, you know, we know there are a number of examples of sensitive periods, yes, both in brain functions and in behaviors. So for instance, you know, this is clear, this is from acquisition of language, yes? We know that our brain is not equally receptive to learn language, yeah, and I'm a living proof of that. You know, the fact that I have an accent, yes, it's an example of this. Now, for instance, we know that once our brain is exposed to one language, yes, in a very short period of time, from 6 months, it's a capacity to discriminate perfectly between sounds of other languages decreases dramatically. Yes? So this is not news. Similarly, another example of a sensitive period is the acquisition of music. Yes? So our brain is not equally receptive to the acquisition of music throughout the lifespan. So learning music below the age of 7 is different from learning music, let's say, at the age of 20. Yes? And there are as well functional and structural differences in the brain of those individuals that were exposed to music early on in life. Now, more related to the subject of the talk today is we also know that experience influences behavioral development. So we have data from animal work, and this is work from Michael Minnie, a researcher at McGill with whom I collaborate closely, that he spent his life studying how maternal behavior in rats measured by tactile stimulation of the mom to the offspring influences behavioral development in the offspring. Yes? So what Michael saw is that naturally rats, they are different in the frequency with which they stimulate their offspring. So there are moms that lick and groom their offspring more frequently than others. Yes? Now, when he looks at those moms that are more maternal, so to speak, that stimulate their offspring more frequently, and look at those that are 1 standard deviation above the mean and compares to those that do this less frequently, yes, and then observes the development of the offspring, what they see is that the offspring are different. The offspring of moms that are more stimulating tend to be more exploratory in their behavioral development. Yes, and this is a stable trait as compared to the offspring of the rats that are less stimulating. Yes, that lick and groom their offspring less frequently, they are more inhibited. Now, what is important is that these behavioral phenotypes correlate, yes, with changes in some areas in the brain and some receptors. So for instance, what they see is that there is a difference in the expression of the glucocorticoid receptor in the hippocampus of these offspring in such a way that the offspring of the moms that are more stimulating have higher levels of the glucocorticoid receptor in the hippocampus as compared to the offspring of the moms that are less stimulating. Yes? All right. So, what we've learned over the last few years is that epigenetic mechanisms help explain how is that this environmental experience influences brain plasticity. Yes? So, I'm glad that you had a grand rounds on epigenetics, so it's going to make my life easier, and explaining to you how these mechanisms work. But basically, epigenetics is the way whereby our genome is modified both physically and chemically. Yes, so it's anything else but the code. Yes, and there are 3 main epigenetic mechanisms. One, which is the change of the of mRNAs or RNA species. Yes, so by other RNA sequences that go and regulate what is transcribed. The other mechanisms is by changing the way the DNA is compacted. So you alter the proteins that are known as histones, yes, that compact the DNA, and therefore you make some sequences more or less available. And then there's another mechanism, which is the mechanism we're going to focus on today, which is DNA methylation. So, what is DNA methylation? Is basically, although this has changed significantly and changes all the time, the central dogma of DNA methylation is that basically DNA methylation is the addition of a methyl group to a cytosine base. Yes? Every time a cytosine is besides a guanine. Yes? So a CG sequence, the cytosine is methylated by the addition of a methyl group. And what is the functional impact of that is that these methyl groups would basically prevent proteins from binding to that sequence and therefore recognizing certain sequences that would regulate the activity of a gene. Yes? So the central dogma is basically regulatory parts of a gene. So you remember, yes, a gene is composed by typically a promoter, which is a region of the gene that regulates the activity of a gene, and then exons, here depicted in the pink boxes, and then introns, there are the spaces between the exons. Yes? So typically a gene originates a protein, and this protein is typically the result of the sequences that are in the exons. Yes? So because the promoter, it's the part that regulates the activity of the gene. So if you add methyl sites to the promoter sequences, and the activity of the promoter is usually it's given by the binding of proteins that would bind there and activate the activity of the gene. If you methylate and prevent the proteins that are known as transcription factors from binding there, the end result is that either this gene does not produce any protein, so it's not activated, or produces less protein. All right? So this is, again, this is the central dogma. It does not mean that it always works there, like this. Yes? And more we're learning this, even for us working in this area, is tough to keep up because information changes so fast. All right. So back to the example of how experience modifies behavior. Yes, so I told you about this work on the rat from Michael's work. So, and I told you that the maternal behavior modifies offspring behavior, and as well correlates with changes in the expression, the quantity of glucocorticoreceptor in the hippocampus of these offspring. Now, one point that is very important is that if you cross-foster, if you take the offspring from one mom that is more stimulating, yes, and you take in a litter of a mom that is less stimulating, yes, and you do this immediately after birth, you completely reverse the phenotype, suggesting that indeed is the stimulation by the mother that is critical, yes, in the definition of this phenotype. Okay, so work on this using this animal model showed that the maternal behavior, what it does, the stimulation by the mom, yes, what it does, basically it changes the amount of methylation in the promoter sequences of the glucocorticoreceptor gene in the hippocampus. Yes? So if the mom rat, by licking and grooming its offspring, yes, it leads to a series of events which end result is a change in the methylation pattern in the promoter region of this of the glucocorticoreceptor of the offspring, in such a way that the more the mom stimulates, the less methylation that there is in this region here of the promoter. Yes? And this region here is important because it's a region where this transcription factor, this protein known as NGFIA, yes, or nerve growth factor inducible protein A, binds. And this factor here binds and gives the signal to the gene that it's supposed to activate. Yes? And by having more methylation here, this transcription factor is enabled to bind here and therefore to give the signal to the gene to produce glucocorticoreceptor. Right? Now, what is interesting and important is that when you cross-foster, again, when you take the offspring from one mom and put in the other litter, you basically reverse the effect. So that suggesting that indeed is the methylation here that accounts for the difference in the expression of the glucocorticoreceptor in the hippocampus of this offspring according to maternal behavior. All right? All right, so why is this important to what I'm about to tell you? Yes? Well, it's important because it shows, yes, that the HP axis, yes, well, I don't have to introduce the HB axis to you, it's regulated according to early environment, at least in rats. Yes? And it's regulated by the early, we can say perhaps, social environment. And by changing the levels of expression of a glucocorticoid receptor in the hippocampus. Yes, so you know that the HB axis involves the hypothalamus, pituitary, and adrenal axis, but also the hippocampus plays an important role regulating the tone, yes, of the HB axis. Now, why is the HB axis also important in psychiatry? But in suicide in particular, it's important because we know that individuals that have a hyperactive HB axis are more at risk of manifesting suicidal behavior. So this is data from Bill Coriel following patients that were admitted in Iowa for many years, showing that those, yes, that were non-suppressors to the dexamethasone suppression test were more likely to die by suicide, by 26% in 15-year follow-up. This is more recent data showing, patients from a primary healthcare area here in the U.S., and I apologize, I don't remember from where, but over 1 million observations of patients that were treated with corticosteroids for medical indications, and then showing how those that were exposed are more likely to show, to manifest suicidal behavior later on. So it was important to us to study whether or not part of this risk that we see in individuals that were exposed to maltreatment early on in life could be explained by differential methylation of the glucocorticoid receptor gene. Because I showed you there's evidence suggesting the HB axis dysfunction increases risk of suicidal behavior, and that perhaps part of this risk could be related to differential methylation. So how do we do that? So we work in collaboration with the coroner's office from Quebec, whereby we invite families from individuals who died by suicide, as well from individuals who died by other causes, like car accidents, like work-related accidents, to donate brain tissue to our bank. It's an ongoing collection, it's an open brain bank, and then the families, after donating the brain tissue, they participate in a series of interviews that we know collectively as psychological autopsies. Yeah, so psychological autopsies are basically a way of reconstructing clinical information from individuals who have already died. Then we complement those interviews with information from medical charts, from youth protection charts. So we have information about their early life development, and information that it's all collected, using structured interviews and interviewing at least 2 different informants per case. So when you look at, the glucocorticoid receptor gene is a gene that it's not easy to understand. It's a gene that has a number of different variants of its exon 1, and these different variants explain its expression in different tissues. One of these variants, it's a variant known as 1F in humans and 1,7, and this is the variant that really is differentially methylated according to experience in the rats. And so this is the area that we focus. And the reason for that is that the 1F is the predominant area in the hippocampus. Yeah, it's the predominant transcript in the hippocampus. So when we looked at individuals who died by suicide and had a history of severe early life adversity, yes, we see first that these individuals have lower levels of expression of the glucocorticoid receptor in the hippocampus, as compared to both individuals who died by suicide but did not have a history of early life adversity, and as compared to controls with a negative history of early life adversity. When we look at the levels of methylation in the promoter of that variant that I told you that explains its expression in the hippocampus, what we see is that those individuals that died by suicide and had a history of adversity have increased levels of methylation in this promoter. Okay? Now, what was interesting is that these increased levels of methylation in the promoter were precisely in the area of the promoter where this transcription factor that I told you before, that it's important in the expression of this gene, yes, that drives the activity of this gene, and the same one that I told you about in the rats, this is where it binds. So that we see more methylation here, precisely where this binds, and this affects the activity of this gene of being expressed. Yes? So I'm not going to bother you with a lot of technical stuff that we did after to show that this indeed is the case. Yes, but the message from this study, initial study, was exactly that. Now, since we've done this initial study, we've conducted other work, yes, showing that in addition to this variant, that it's important in the expression of the glucocorticoid receptor in the hippocampus, other variants of the glucocorticoid receptor are also differentially expressed in the brain of individuals who died by suicide and had a history of severe maltreatment of the glucocorticoid receptor, but not everywhere in the brain. Yes, so clearly in the hippocampus that's the case, but in the cingulate gyrus we couldn't find those differences. Yes, so we're still now trying to understand, yes, precisely where those differences are important in the brain in terms of driving the expression and of the gene. So since we initially published the work, a number of other groups have looked at similar effects, yes, and the results so far look very consistent, yes, but of course, it hasn't been that long, and we're still we're still to see exactly what comes out. But there are reports both using peripheral models of this, in other words, looking at peripheral samples, yes, of individuals that were exposed to adversity, and comparing with controls, show increased methylation of the GR gene in the same region of the gene of the 1F variant, or as well looking at both in clinical samples as this one or in cohorts, showing as well a relationship with the expression of the GR gene peripherally, as well as with other phenotypes related to adversity and as well in the brain. Yes, so this, as I said, is still early, but I think the results are certainly encouraging. So after we've done that work, yes, the question we asked, well, this was a more hypothesis-driven study, yeah, based on the data from the animals and based on what we know from work in individuals with suicidal behavior. Now, the other questions, maybe there are other genes as well, besides the GR, that are differentially methylated. Yes, so we've done a study using a genome-wide approach. So in other words, an approach where you look at the whole genome and using a technique known as MDG. Yes, so basically what you do here is you use an antibody to detect regions of the genome that are methylated, and then you hybridize an array that contains sequences for all the genes in your genome, and then you look at intensity of the signal. So when you would do that, what we see is that, well, first, there's no tendency to be differential methylation in different chromosomes, yes, as you would see, let's say, in cancer. So this is just equally distributed across the genome. We saw 355 differentially methylated probes in this study that focus only in promoters, yes? And of these, 245 that were hypermethylated and 110 hypomethylated. Now, because we had, and this is in the hippocampus, yes, again, because we had on these individuals as well data on expression levels of all the genes in the genome, yes, that was collected using mRNA arrays, so arrays that look at signal from activity of the genes. We were able to put together data on methylation and expression. So what we see is basically that, as expected, the more methylation, probably you won't see, and I apologize, it's a small slide, but at the more methylation in red, yes, the less expression in green, and vice versa. Yes, so suggesting that indeed at the genome level you have an overall relationship between methylation and activity of the gene. Yes, look at the p-value here. Yes, I don't think I ever had a p-value as significant as this one. Yes, it's 10 to minus 247. Yes, it cannot be more significant than that. Yes. All right, and what is that we see when we look at differential genes? Yes, that are differentially methylated. So, this is a list of the genes that are differentially methylated according to real life adversity, yes, in these individuals. And but what I want to draw your attention is that even when we do this study using a non-hypothesis-driven approach, yes, it was very reassuring to see that the glucocorticoid receptor comes out as one of the most significantly differentially methylated genes, yes, in the whole genome. Now, when we look at all these 200 and something differentially methylated fragments, yes, and group them according to function, yes, it was also interesting to see that among the different, we call these gene ontologies, but basically among the different functions of the genes that are differentially methylated, we see genes involved in neuronal cell and neuronal plasticity. Yes, so basically suggesting that indeed, when we're looking at genes that are differentially methylated according to our life experience, yes, it seems to me that it's not just by chance, yes, that we have a lot of genes that are involved in how our brain reacts to experience. Now, you're going to ask me, but okay, you're looking at the brain and we're looking at homogenates of brain tissue. So basically what we take is it's a sample of the hippocampus, yes, and they will homogenate that sample and we look at patterns of methylation. Yes? But perhaps, when you discuss about methylation, one of the points that you probably discussed is that methylation is tissue specific, yes, or so we believe, that it drives how, we have the same DNA in all our cells. Nevertheless, the cell from our brain, the neurons, don't produce the same thing as our hepatocytes, yes, and don't function the same as our osteocytes, yes, but they have the same DNA. So what determines the difference in phenotype of a cell if the DNA is the same? We believe that to a large degree are epigenetic processes that are related to the differences in phenotype of a cell, what it does produce or not. So a pattern of methylation in a neuron may be different from a pattern of methylation in an astrocyte or in an oligodendrocyte. So what we do is are interested in looking at the different cellular fractions. So how we do that? So we use a technique that's known as FACS, yes, whereby we use an antibody to select DNA from specific cells and separate those that are from neurons from those that are not from neurons. So when we look at that, yes, so this was alcine, which was the gene that was the most significantly differentially methylated in the list of the genes that we had in this genome-wide methylation study. When we look at each pattern of methylation, we see that we had more methylation among those individuals that were died by suicide and had a history of abuse, as compared to those that were died by suicide but were not abused, and the controls. We had increased methylation here, and this increased methylation here, when we artificially methylate a cell model in this region, we show that the activity of the cell in the production of this gene is decreased. Yes? So what I wanted to show is that primarily these results were being driven by the neuronal fraction and not by the non-neuronal fraction, which is here. Yes? That you don't see a difference when you look at the non-neurons. That this difference in methylation was primarily observed in the neuronal fraction. And so we look at all of the genes that were differentially methylated, yes, among the top genes, and it was perhaps coincidental. I mean, we didn't look at all the 240 because it would be a tremendous amount of work. But we looked at the top 10, and most of these signals were being observed in the neuronal fraction as opposed to the non-neuronal fraction. Now, not to say that the non-neuronal fraction may not have methylation patterns that are specific to them and may drive this effect, but I just thought it was interesting to observe that this effect was driven by the neuronal fraction. So, what can we make out of this? You know, focusing on the GR, is that this is the hypothesis that we have. Yes. So individuals that are exposed to a life adversity, yes, as I showed you, have increased methylation in the hippocampus of the GR gene. Yes. So I told you that if you have more methylation, your gene is less active, you have less expression of this gene. If you are less active, have less expression, therefore you have a decreased number of receptors, yes, of the glucortical receptor in the hippocampus, as opposed to individuals that would have just a normal development. So as you know, if you have a situation of stress, yes, it would activate the HB axis to produce cortisol that would then have a negative feedback on the hippocampus. And how this negative feedback occurs is that the cortisol would bind to the glucortical receptors in the hippocampus to let it know that it needs to decrease the activity of the HB axis. But if you have less availability or less amount of receptors in the hippocampus, your negative feedback is less effective. So if it's less effective, you're basically going to have an overactivity of the HB axis. So this is what we think happens in individuals that were exposed to adversity, is that they have, as a result, an HB axis that is more active. Now, I don't want to bother you a lot with this data, but this is just comparative data that we've done looking at humans and rats. Yes? And just showing that there are a lot of parallels between the species, suggesting to us that these are mechanisms that are evolutionary conserved. Yes? And why would that be? We don't know, but we can speculate that perhaps these are important in helping the organism to better adapt to the environment. Yes? So the idea that we have is that all these mechanisms, yes, are adaptive, usually. Yes? So they would help the organism better respond and adapt to a hostile environment. Yes? So it makes sense if you think that if the environment is hostile, what you need to be is all the time alert. Yes? How can you be all the time alert? You need to be all the time alert. You have to have an HB axis that is more active. Yes, as opposed to another situation. So this would be an adaptive mechanism in an environment that is constantly hostile. The problem is that when you moved to an environment that is not so hostile, that mechanism that was adaptive at one point becomes maladaptive in another environment. Yes, so that's what basically we think that may be behind this. So along those lines, you know, in the last few minutes, what I'd like to discuss with you is how is it these can translate into suicide risk, or a risk, an increased risk of suicidal behavior. And so back to the cohort. So this is a cohort representative of the population of Quebec. Yes, so of 3,000 children that were recruited to this cohort because they were attending kindergarten, French-speaking kindergarten across the province. Yes, and so 1,500 boys and girls. And then from age 6 to 12, every year, the moms and their teachers in kindergarten and then at school were asked questions about the child, about their levels of anxiety, their levels of hostility, aggression, hyperactivity, etc. And then later on, we have information on their psychiatric diagnosis and a number of other things. So it's a wealth of data on this cohort. Yes? So the beauty of having these sort of longitudinal studies is that you can look at not only 2 points in time, but rather, you know, over time. Yes? So first, when we look at, in this cohort, as I showed you initially, children from this cohort that were exposed to adversity early on in life are more likely to manifest suicidal behavior in the forms of suicide attempt when they grow up at the age of 21. Now, because we have this longitudinal data, we can build trajectories, yes, of behavioral development. And so, you know, we're talking about the GR gene. I suggested that it has to do with being hyperactive. So at the clinical level, what it means is basically anxiety traits, isn't it? But that's what anxiety, to some degree, is. You know, it's being all the time vigilant, hypervigilant. So, what we see is that children from this cohort, that from age 6 to age 12, are systematically, consistently anxious. Yes? As opposed to children that are either not anxious at all, or were anxious and decrease or increase, membership in this group increases once controlled by all the factors. The likelihood of manifesting suicidal behavior later on in life by 60%. Yes? Okay, so now one more piece of information that I want to give you before we put all this together is that important, I think, to this discussion is that often when we look at individuals who die by suicide, and this is a study we've done, you know, many years ago, looking at individuals who die by suicide in the context of an episode of major depression and compare them with depressed controls from the clinic, we see that these individuals have a tendency to have more impulsive traits as well as more traits of hostility and aggression. So, when we look at the relationship between, in the cohort, yes, of having been abused during childhood and manifesting suicidal behavior at age 21, and we look at children that had externalizing disorders, yes, we see that the relationship between having been exposed to adversity and suicidal behavior is fully explained by the development of high anxiety trajectories, yes, at the statistical level. So it's fully mediated, yes, as we say. Whereas among children that don't have externalizing disorders, what we see is that the relationship between having been exposed to adversity early on and manifesting suicidal behaviors remains partially explained, but not fully, by the development of high anxiety trajectories. All right? Okay, so how can we speculate, yes, and this is really speculation, I don't have much data for this, but speculate that this can translate into an increased risk of manifesting suicidal behaviors. So what we think is that many times individuals who attempt suicide, they have an altered attribution of emotional value to certain acts, yes, in social interactions. And that this leads to an impaired emotional regulation. Yes, and we think that having an overactive HB axis somewhat acts in this path here, yes, by making it more abrupt, yes, or by facilitating acting on suicidal thoughts. So what supports this relationship? Well, there's data, for instance, suggesting that individuals who attempt suicide have impaired decision making. Yes. And there's a lot of, I think, data on this level. Also, we've done studies looking at relatives of abused suicide completers, yes, and first-degree relatives, siblings, yes, that likely were also victims of adversity like the individuals, and showing that when they are subject to the Trier stress social test, is that it's a psychosocial stressor in the lab. There's marked difference between how they react to a stressful event as compared to the controls. Yes, and there seems to be alterations in executive function that the exposure to the stress is differently processed between them and controls. So, in conclusion, yes, what I discussed with you was that a portion of individuals who die by suicide have a history of early-life adversity, that there's data suggesting that epigenetic mechanisms, and we talk about DNA methylation today, may explain this increased risk. And I gave you one example of how this may happen, which is through hypermethylation of the glucocorticoreceptor gene. Yes? I want to stress that this is what aspect of suicide, yes? That most of the individuals who died by suicide were not exposed to life adversity, yes? But nevertheless, a significant minority does, and I think it's important for us to understand mechanisms involved with this. Now, these are distal risk factors, yes, that play a role, so we think, by increasing predisposition. But there are a number of other proximal risk factors, none of which we discussed today, that are very important, yes, in understanding suicide risk and suicidal crisis. All right, so the work that I discussed with you is work done by a lot of different people. Yes, in particular, the work on the glucocorticoreceptor was done by Patrick McGowan, who was not in the picture here, who was a postdoc at the time, working with us. Benoit Labonté did the work on the genome-wide array. Naguib Meshauer, with whom I share the responsibilities of the brain bank, and a number of other people that were or have involved and left the lab before the picture. They were involved in aspects of this work that I won't be able to acknowledge now. In addition, I mentioned to you Michael Meany, with whom I collaborate in a lot of this work, as well as Moshe Schiff, and from the cohorts, all the works of the cohorts, it's in collaboration with Richard Tremblay. So last but not least, I just want to take 1 minute of your attention to advertise a conference that we are organizing in Montreal in June this year from 10 to 13, where we're going to be discussing research on suicide, and this is a conference in partnership with the AFSP. So thank you very much.
I was wondering, one of the interesting pieces of the epigenetic mechanisms is that they get, they can be inherited. And I'm wondering if there's any plans or if there is any research that's been done on whether those, say those cohorts of folks who've been abused but didn't experience abuse themselves, if they had those same, DNA methylation profiles of the glucocorticoid receptor and how that sets them up for it.
Yeah, so that's a very important question and timely as well. And I would really, I would caution in taking conclusions about the inherited capacity of methylation or epigenetic mechanism. So this is a very contentious issue, yes, whether or not epigenetics or epigenetic factors are inherited. So the knowledge until now has been that these are not inherited, yes, or inherited in the sense of transmitted from one generation to another. Yes, they are inherited from one cell to the next cell. So when a cell divides and divides and generates two cells, yes, the epigenetic marks of the parental cell are transmitted to the other cells. Yes, but not through meiosis. Yes, or not through. Or that has been the wisdom. Now, over the last few years, there has been data suggesting that epigenetic marks may be transmitted from one generation to another. Yes. There are a few examples of these transmission. Yes, for instance, I think the famous one is the Dutch famine, for instance, and how one generation that was exposed to adversity, how the second generation gained some of the traits. Yes, but there's no molecular evidence that that happens. Yes. So there is now more and more work in animals addressing this issue. So my reading, yes, and I'm sure other people may disagree, but my reading is that it's too early, yes, to say that these marks are transmitted. Yes, in part because there's a lot of evidence from embryogenesis that the blast, yes, when you have a new organism being formed, yes, goes through the embryogenesis phases, that if you take a cell, or if you take a blast, let's say, after a morula, that all the epigenetic marks have been erased, yes, and they are then remade some according to parental origin of the of that particular chromosome. So there's a lot of debate as to if it is, what it is, and if it does happen, what is exactly transmitted. You understand that if this is the case, we have to revisit all the theories of evolution, yes, because the way we thought until now was that we evolved not by acquiring genetically or in our genome things like that. Yes, so that would give much more room for Lamarck, etc. But so it's very, very contentious. Yes.
Certainly, this is a really impressive body of data, and you made such a great argument for the impact of abuse and methylation. What I'm wondering is if we have any data about demethylating those areas. You know, we'd like to believe some of the tools or therapies can have an impact, and you're showing very convincing data that suicide risk is elevated. Can we reduce that? Are there things that we can do directly or indirectly? Do we have any evidence?
I think it was a fantastic question. I wish I could answer.
Not yet?
We don't know. We don't know. I think that, there are many more questions than answers that we can provide in this area, yes? So certainly, epigenetics is a very young field of investigation, and it's a very challenging one, precisely because of all the things that I said before, is that they are likely, the changes are likely to be cell-specific, are likely to be tissue-specific, etc. So we know that we can change. Yes, that we know. We know that we can change epigenetic marks. For instance, pharmacologically, we can change both methylation as well as histone modification. Actually, among the drugs that we usually use, like valproate, for instance, is a classical HDAC, as we say. It's histone deacetylase inhibitor. So what it does is it activates more the activity of a gene. So that's, with valproate, is a drug that we use routinely in psychiatry. We can also use drugs that would change methylation. Yes, the problem is the drugs that change methylation would change all methylation. Yes, all methylation in all the areas of the genome, in all the cells. So what you're going to have is basically a disorganized function of the cell. Yes. So the ideal in the future would be, yes, for us to have a way to selectively target certain cells, yes, let's say in the brain, only those cell types, yes, and only that particular sequence in the genome of the cells, yes. So that seems like a huge challenge right now. Yes, it's not impossible, but it's a huge challenge. What I meant by I don't know primarily is, can we change these patterns by, let's say, reconditioning? Yes, for instance, by therapy, by relearning and reprocessing. I don't know. Most likely yes, but I don't know.
For instance, a simple thing, simple for me to say, experiment if in the rats who didn't receive maternal grooming at the high end, instead of fostering them as pups later in life, after we presume it's a sort of fixed pattern, is there any remediation that you can see in terms of their genetics of that?
So in the rats, yes. So in the rats, we can, once you establish the methylation in the rat, you can, that is stable. Yes.
We haven't found anything that changes that.
No, we can change it pharmacologically. We can.
Yeah.
But the problem is, what I said, the rat, you can just add a drug that would remethylate everything.
Like for instance, if a handler,
yeah,
mimicked something they didn't get early in life, and I'm thinking sort of the parallels to what we do in therapy, does any of that turn out to be significant later? It may not be, I don't know.
No, we don't know. So, in the rat, we know we can manipulate this pharmacologically. But, as I said, it would be unthinkable doing something like this. Because you completely make the system dysfunctional.
Well, I just want to say, too, I would have to believe, to some extent, that we may not have to be as, how do I say this? Perhaps some of the interventions that we do that work will turn out to be doing exactly what we would predict. And we don't have to manipulate the genetics to get it right at a different level. But the nature of the being will take care of it in the way that we would.
Yeah, absolutely. I just don't know.
Right.
Yes, whether that's happens or not. I mean, I think that it's likely that that happens. You know, that's basically what resilience is about. Yes,
Exactly.
most likely.
Right.
But I cannot tell you that that's because, you know, well, first of all, we work with postmortem brain tissue. Yes, we don't get the postmortem brain tissue of those individuals that are resilient. Yes. And then the other thing is that what I said. This is very in the beginning. Yes. So there's still a lot to learn. Yes.
I read an article in which Michael Meany, with whom you work, it was said in the article that it was said by him that parental nurturing can counteract a lot of this early methylation. Now, I don't know if that's anything that makes sense to you, given that you know him and you work with him, but that also picks up on this issue.
Yes.
What you can do to counter methylation.
So what he's talking about is that I imagine that what he's talking about is that in the rat, what you can do is you take the rats from low licking and grooming mums and you put them in environments that are highly enriched,
Right.
and you reverse the behavioral phenotypes. Yes? So you take these rats that are more inhibited and you put them in, in cages that they can run and can do things, socialize, interact, and then these rats become resemble the phenotypes of the high lickers and groomers.
Right. And then you wonder if there's a window within which you can do that to make a lasting impression, and maybe other times you can't.
Yes. So typically, it's really early on in life. Yes. So in the rat is in the first weeks that you can change and manipulate it and modify the activity of the HB axis of the rat.
Are you making a distinction between different kinds of adverse events? At one hand, you're talking about maternal grooming, and then also about abuse, physical abuse, and they're not the same thing, obviously. So were you just sort of taking a global approach to these events, or were you making any kind of distinction?
Yeah. So let me be very clear here. The animal data, I'm pointing out to it because it's the first example that we had in which social interaction or social environment regulate the activity of biological systems. Yes. I'm not making a parallel between that and the abusive experience per se, other than to say that the HB axis is regulated and adjusted according to our life. For the human work, we really focused on extreme phenotypes. Yes. So the abused individuals were the higher, those that score the highest in the SICA, which is the instrument that we use to characterize this, which is an instrument it's a structured instrument to collect information on life accuracy. And then in the study, so we took those that have the highest scores in that instrument, and together physical, sexual, and neglect. Yes, but what you need to understand is that these were individuals that had histories that were really, really very, very, very tough. Yes, so these were individuals that typically were taken away from their from the custody of their parents. Yes, put into foster care, and then endure in foster care as well a series of abusive experiences. Yes, so these represent the group that was studied here.
I've got a lot of thoughts about this and trying to put it in some context of other things we've learned. And I can imagine there was an extraordinary excitement as you worked at this methylation hypothesis, because you tied together other data from previous eras, like the 2001 Coriell dexamethasone non-suppression and the high risk. and you began to find a way to see how that whole thing was plausible mechanistically. And it must have made you really happy when you actually did it artificially and methylated and saw lack of production of that glucocorticoid receptor. So I can just imagine a lot of excitement as you tied together naturalistic data, the dexamethasone non-suppression, and then your own manipulation. You got that awesome p-value you showed us. Were you guys ecstatic when that happened?
So, you know, to tell the truth, you know, I wish it was the way you described it.
Now you can put it that way. Yeah.
In reality, it usually doesn't work this way.
Yeah.
So we have findings, and then we break our heads to find, you know, why is it, you know, why is it we have, and why did it make sense?
Right.
You know, we also work, you know, we have hypotheses, etc. But, you know, in our case, so, you know, why I got into this business was because, so we were working primarily with transcriptomes. Yes, so my work was primarily focused on trying to understand what changes in terms of function in the brain of individuals that feel hopeless. Yes, so hopeless that the only way out they can think is killing themselves. So we're working with looking at the transcriptome in the brain, and then as it happens, I worked. You know, my committee works in the same building that I do. Yes. But you know, that's how life goes. Yes. So you work in the same building, you never talk. You know, you never have time to. You know, all busy, and you know, you see, hello, how are you? This and that. Yes. So it took me to one day, you know, to go to SOBP, which is Society of Biological Psychiatry, where he gave a talk to sit down and see exactly what he was doing. Yes. And there, you know, far away, and then I, you know, said, well, you know, suddenly that made sense to me based on what I knew. And then I said, well, you know, we have to do this as well, you know, in our work. Yes. So that's how our collaboration began. And that's how the translation from the rat to the human began, because we had the human data, you know, the capacitors set these in humans, and he had experience looking at these in the rat.
So I have a few more questions I want to fill on that. Last year, Victoria Rondo came and spoke with us and gave a wonderful presentation. And the gist of it was, so she taught us many things, but one of them was when you looked at the production of serotonin in the radionuclide midbrain, there was plenty of it there, but she was beginning to think that the issue was the axonal transport to other areas of the cortex and the limbic system was the problem. So she said, now I have to, after all this lifetime of work, I've got to study axonal transport and look for why that serotonin can't get distributed in the brains of people who commit violent suicide. So when you think about that work, there's another researcher devoting her life the deepest way as you have. She's working at a different angle. And when you think about that, you think about that as sort of you and your colleague sort of working in the same building but on separate paths, or do you see some connection between those two things?
Yes. So the work that Vicky does is extremely important, and it's, you know, it's an area of work that has really made a huge difference in the way we understand biologically individuals who die by suicide or have suicidal behavior. The way I see this work is that it's complementary to the work that she does. Yes. Now, how they inform each other, we still don't know very much. Yes. Certainly, the brain is a system, you know, that at least I usually think of it as a system of communicating pipes, you know, where, you know, you change something in one area, the other thing, you know, goes up, etc. So and the systems at some level are interconnected. Yes. So her work, you know, has focused really on serotonin and the decreased serotonergic hypothesis of suicidal behaviors. Now, very possible that epigenetic factors play a role in explaining the function of that system. As a matter of fact, we have data. You know, I presented what I thought would be more clinically relevant, but you know, we have a lot of data showing how the serotonergic system as well is differentially regulated by methylation according to our life experience. Yes. So there is an area of in between the monoamine oxidase genes. Yes, they're both in chromosome X. That is differentially methylated, and that area has a major impact in how MAOA is expressed in the brain. So this could be, you know, one way of putting this work together.
Another question about her work was, if I got it right, she used a brain bank of people who died by violent suicide, who had never been medicated, no overdoses. So she wanted to have a really clean, violent suicide group. And I was wondering, in your brain bank, do you have a similar sort of inclusion-exclusion criteria, or do you have a different kind of patient in that brain bank?
So, you know, in the brain bank that we have all types of brains, yes. We all want to, as much as possible, control for possible confounders and effects, yes, that would confound, you know, the results that you have, yes. So, but at one point, yes, it is obviously very difficult to completely control for everything, because we're studying humans, not rats. Yes, if you want to control everything, go on and study rats. Yes, but humans, you know, as much as you can control, you cannot control for all diversity. First of all, you only have information that you have access to, all the information that maybe you may not have access to. So all this to say that we all want to do that, and we do as well. We try to end up with as much homogeneous groups as possible. That said, you know, individuals, this profile of individuals that I presented to you, you may easily understand from a clinical point of view, these are individuals that are likely to have a lot of morbidity. So there are individuals that often have histories of substance abuse, and etc. And it's very difficult to end up with a group of individuals that are exposed to this level of diversity that will be clean from everything. So the way we do is that we include for these studies, all the studies we separate. For these studies we include more heterogeneous groups. And then we control for these different factors, either statistically or we use animal models, yes, where we expose the animals to substance, and then we look at how they will or not affect the respective fibers.
One more question is about, we're training medical students, residents in psychiatry, and all of us who are practicing psychiatry, continually trying to enhance our risk assessment, our study. Do you take from this work, what are some of the principles you might translate as you move back into your clinical role for us to be thinking about as educators in risk assessment of people we're treating?
Yeah. So, well, that's a very good question, and I'm not sure I'd be able to give you a clear answer about this, because you know, when you look at risk from the clinical point of view, yes, what basically you need to do is use all information that you have available to draw, you know, make best informed decision. Yes. So these type of at least the molecular information that I gave you is not of use at this time. Yes, to help you in making a clinical decision about risk. So let's say if you are in the emergency room trying to assess the risk of someone, then that information is not going to help. On the other hand, it may help you, to better understand mechanisms, as well as I think that what will help you is the second part of the talk, yes, in terms of understanding risks and traits that increase this risk. Yes. So that would help you. But clearly, this is not the type of information that would be extremely useful when you're at the emergency room at this stage.
This seems to be helpful and explanatory way to patients. It's
Yes.
understandable how you may have this anxious system driving you in this way, and we're going to try to do something to help you with that.
So, when we published this initial study, which was the first time that someone, had shown that exposure to trauma in the life had biological impact on the brain. We were overwhelmed by patient groups and advocates that were extremely happy, yes, with this finding, and felt happy in the sense that we gave them some meaning, yes, to a lot of the difficulties that they had faced over the years. And so I think in that sense, it certainly is helpful in terms of validating.
You had a question.
I'm Nita Sharma, one of the psychiatry residents. I do mostly right now in outpatient work, and this is a clinical challenge. After listening into this, it's very good to know about the epigenetics and everything, and I'm thinking, how do I use it in my clinical practice? About the exogenous steroids, like if we have a profile of a patient who matches the criteria of childhood abuse, who's always anxious, and really she was on a long-term steroids because of massive pericarditis, 3 months, and now it has been tapered off, and I see her anxiety going off the roof. That's kind of how do I use this clinically, and how do I help my patient?
Well, so now this, if I understand, your patient has been on steroids because of
Pericarditis.
yes. So I think what you need to do is really work within the medical indication that there is for the steroids, and then taper them off very slowly. I think that would be a way for the system to readapt, as much as possible. But it's something that you need to discuss with the internal medical team.
So is there a correlation of exogenous steroid to that HPA axis when already their HPA axis is at a very high alert level, and then they get steroid and then they taper it off? And
So you have to see on this particular patient, because some patients when you have, so obviously if you have exogenous steroids, then you would have an impact on the activity that would be suppressed most of the time, yes? Not in everyone, so then sometimes it's compounded. So it's difficult in that particular case without knowing the details.
Would it increase the suicide risk? And I'm worried, what would be, how can I help her in outpatient?
Yeah.
We cannot send her inpatient and for this particular.
So at the statistical level and epidemiological level, yes, it would increase. Yes, so that's what this study that was published last year in the American Journal shows very clearly how patients that were prescribed with steroids for other, corticosteroids for other, for medical reasons, yes. And this is based on a very large sample, almost 1 million observations. They had an increased risk of those that were not of manifesting suicidal behavior. And among all the psychiatric phenotypes or symptoms, or illnesses, it's suicidal behavior that was the most, or the strongest, or had the strongest effect. So from that point of view, yes, but does not mean individually, yes, that that's the case.
Thank you.
Okay.
Would it work, I mean, just trying to apply the model, maybe it doesn't work, but if they were getting exogenous steroids, they probably suppress those hippocampal glucocorticoid receptor production by having so much available, right? So that when you taper them, you'd have to taper them even more slowly and pay attention to suicide risk as it evolves, or?
No, because what happens is that you have a negative feedback, so you decrease the activity of the HPA axis. Yes.
Okay.
So basically, if it functions normally, basically when you have exogenous steroid, it would activate the glucocorticoreceptors, in the hippocampus. I mean, there are receptors in all areas as well. It would decrease the activity of the HPA axis. Once you taper off, then normally the system will come back to normal. Yes. The point is that if you have more methylation, that's what I was making here, so then we have less availability of glucocorticoreceptors. So that's constitutional.
Was your, and I may have missed this in my understanding, basic understanding, but was your impression or is your working theory that the initial cause of the methylation, the increased methylation, is because of the stress leading to an increased kind of cortisol or adrenaline usage in the body? Like, is it because of that actually, which would make sense out of what Nita's asking as well?
So you didn't miss it because we don't know.
Okay.
Yes? What, we don't know what is that the relation between having been exposed to the stress early on in life and what are the molecular mechanisms that lead to increased methylation. We have some idea about this in the rat, but we don't know in humans.
I mean, it seems like it would make sense in terms of evolution if the actual downregulation of those receptors came because we were using it too much and they were like, whoa, 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 we should slow down the system. I mean, it would kind of make sense, which would, again, also...
It could be. In the rat, it goes through a different path. Yes, it goes in the rat because it's through tactile stimulation. Yes, that it happens. It is the tactile stimulation that, because you can mimic in the rat the same thing by, it can be a mum, or it can be you just get a brush, yes, and you stimulate the skin of the offspring, and you have exactly the same effect. It doesn't have to be the mum, because in the rat it's really the tactile stimulation that works. And that tactile stimulation releases serotonin that stimulates thyroid hormone, and that leads to the activation of an enzyme that demethylates that region that normally would be completely methylated. So the more you stimulate, the more the methylation decreases. So that's the mechanism in the rat. I don't know in humans what it is. Yes. I mean, I don't know in humans. I don't know in humans a lot of things. One of the things I don't know is what is a critical period? Yes. So is it until 12? Is it until 14? Is it longer? Is it what is the experience? So I don't know. I can only do these studies retrospectively, as you imagine. Yes. And so all these temporal relationships are impossible to measure.
One other question is, in the total population of people who die by suicide, is this part of it, the low glucocorticoid receptor, the early adverse experiences, does that account for about 10 to 40%? Or what percentage? Then you have a whole larger group that this explanation isn't applicable to. Is that right? How would you think of that?
So, so this group that we looked at, these factors represent a small proportion, yes, because these are those that had a history of adversity, and among those that had a history of adversity, those that are more severe, yes. So, these studies are difficult to do, are expensive, and so you need brain tissue, etc. So we cannot generalize that these effects occur at everyone. But as I mentioned, when you look at history of adversity in individuals who died by suicide, we see these, depending on how you define adversity, anywhere between 30 and 40%. Yes, so 40 is more liberal, and 30 it's less. Then if you look at very severe, it's a smaller proportion. So that answers your question. But I don't know if these mechanisms play a role in everyone.
I just have a quick question about the slides you had with the neuropsychological instruments with the Gels Kaplan. So did you say that in those studies that people were exposed to a certain stressor and then their performance on trails and the Stroop test went down in reaction? Is that what you?
That's right. So what that is, this is known as the Trier's social stress test.
Okay.
So I don't know if you're familiar with this. I should have explained. So this basically is that you ask. It's a psychosocial stress in a lab. Yes. So you ask a subject to come to a lab, and you mislead them. You tell them that they will they're going to go through just an interview. And then the way it works is that they are asked to stand on a stage. And there are two members of the lab dressed in a lab coat with no facial expression that suddenly ask the person to start talking about themselves, yes, and then ask them to stop and start doing a series of mathematical operations that are very difficult to do. Yes, so invariably everyone makes mistakes. Yes, and every time they make mistakes, they ask to start again, and they are told, well, you're making errors. Yes, try not to make errors, and every time, so it's a stressful. It releases cortisol. Yes. Obviously, after we debrief and tell them, but there's no other way that you can do this in a lab, So, so we've done this test and did this neuropsych test before and after the stressful situation, measured cortisol. So we see that there is a deficit, yes, or a deficit maybe, but there's a difference in learning process between the groups and the comparisons. So that the comparisons, when they repeat the same test, a few hours after, they improve significantly their performance, whereas they don't. They actually
exposed to stressors. Yeah.
The exposure to stress somewhat alters
Yeah.
their capacity to learn with the experience.
Thank you for a wonderful, wonderful